Hi everybody. I thought I'd show you a picture of my face for a change just so you can see that I'm still alive. And I thought for these videos I'd wear a different hat every time I did a video. This is one of my favorite hats. It's a Stormy Chromer, which is kind of unique to the UP. I like it especially because it's green, so it's Northern's colors. So I hope everybody's doing well, and then I hope the videos are helping with the understanding. Let's go over the answers to the worksheet for chapter 11. So here you see the worksheet. You don't have to write any of this down because the answers to the worksheet are already posted on Educat, but I thought I'd go over a couple extra things on this just to clarify some things. And instead of putting the letters for the word bank in the boxes, I actually wrote the appropriate name or molecule. So let's just briefly go through this. There are 4 colored shapes here that represent oxidative phosphorylation or the electron transport chain. And there are 4 complexes, 1, 2, 3, 4. They're the 4 colored shapes. They're just in numerical order, left to right, 1 through 4. And the role of those transmembrane proteins is to take the electrons from either NADH, the reduced coenzyme NADH, or FADH2, and those electrons then are both handed off to coenzyme Q, which is here. So coenzyme Q grabs electrons from NADH, becomes the reduced form, which is written as QH2, and then QH2 hands the electrons off to complex 3. The same thing happens with FADH2. It hands off its electrons, becomes oxidized. Those electrons go to complex 2, which hands them to coenzyme Q, making the reduced form, and they're handed off to complex 3. Then at this point, cytochrome C, which is not a transmembrane protein, but tightly associated with the membrane, will take the electrons from complex 3 and carry them to complex 4. The final electron receptor is molecular oxygen. That will take the electrons from complex 4, combine with hydrogens, and make water. So that's your final reduction of the whole electron transfer chain. The final colored shape here is ATP synthase. It has a transmembrane domain, a catalytic domain, and its function is to take ADP and PI and convert those to ATP. This is the F0 domain, and this is the F1 domain, but the correct answer for this is ATP synthase because it's in this part of the molecule, the box. All right, so the other thing you should notice is we're pumping 4 plus 4 plus 2 hydrogen ions across this membrane. If we start with NADH, we pump 4 plus 2 if we start with FADH2 because it starts later in the chain. Those protons build up out here in the intermembrane space and create a pressure when they come back through. They come back through the ATP synthase. That pressure is relieved. You can consider that pressure as a potential energy, and when that pressure is relieved, that energy is used to synthesize ATP. That process is called the chemiosmotic theory. All right, let's look at the other parts of this worksheet. So this one says, which complex has the lowest reduction potential? That would be complex 1. The other way I could ask the question is, which has the highest reduction potential? And that means the most positive reduction potential. If it said highest, it would be complex 4. So lowest is 1, highest is 4. It goes in this order. We pump hydrogen ions across the membrane. We just talked about that. And they come back through the F0 subunit of ATP synthase. So we can just label those. This is the F. It's actually FO, not F0. And this is F1. The coenzyme that yields the most ATP in the process is NADH because ATP is directly related to how many protons you pump out. NADH provides the pumping of 10. FADH2 only provides the pumping of 6. So NADH would be the highest. An example of a group or cluster that passes the electrons would be the iron-sulfur cluster. So what we're looking at is some group on one of these complexes that actually handles the electrons. And I have a picture of that here from your textbook. So here are 2 examples of iron-sulfur clusters. that are incorporated into some of the complex proteins. The sulfurs are the brownish spheres and the irons are the teal spheres. So you can get different shapes of these, different amounts. This is a diamond shape and this is a cubic shape. The last question on this part of the worksheet is what translocases are not shown in the picture but absolutely essential, and they are for phosphate and ADP. So remember, if we look at the ATP synthase, ADP and P are reactants, so they have to be presence in the matrix here in order to have this reaction catalyzed. So I have a picture of those as well from your textbook. So it's this figure 11.35. So there's an actual translocase or transmembrane channel protein that allows phosphate to come into the matrix. Also, a proton comes in every time you bring in a phosphate. And there's a separate translocase that allows ADP to come in. So both of these will enter the matrix, be converted by the catalytic part of ATP synthase to make ATP. The ADP translocase will also allow ATP to go back out of the matrix, so it's actually called an ATP/ADP translocase. The only other thing that is of note in this picture is notice one proton comes in through the phosphate translocase, and then 3 protons need to come in to make 1 unit of ATP. So the total number of protons you need to make an ATP is 4. So let's go back to the previous picture, and this shows you how we get the amount of ATP per reduced coenzyme. So for NADH, 1 molecule of NADH, you get 10 protons pumped into the intermembrane space. 4 plus 4 plus 2 is 10. You need 4 total protons to make an ATP, so that's how we get 2.5 ATPs made for every 1 NADH. And then FADH2 is less because we only get 4 plus 2, 6, because that starts here. So 6 protons, again divided by the 4 per ATP, equals 1.5 ATPs made from every reduced FADH2.